Good morning, everybody. I enjoyed the presentations yesterday, learned a lot about your myrtle rust and everything. And back in 2006, I was asked to speak. I was asked first to put together a white paper with some other people on sudden oak death. And then I was asked to speak at a conference, kind of like this, but it was a oak mortality task force meeting in California. I'd never been to one of those before. First thing I said when I got up there is, I know less about sudden oak death than anybody in the room, and I'm not a pathologist. I said, but it doesn't matter because genetics and breeding, it works the same across pretty much all species and common understandings and stuff. And so I think a lot of the same principles apply here. I've been involved in resistance breeding since 1991. I organized for forest trees a bunch of international conferences, et cetera, too, mostly because I want to learn to make our programs better. And when I first started at my facility, Dorena, there was a meeting that was held every 2 years called Rust Busters. And it was a meeting of researchers and those in practical realm to talk about the issues between the different regions and between the US and Canada in terms of how to advance programs and to debate the issues. So with that, we'll go ahead and get started. Let's see if I can figure out this device. Okay. So what I'm going to talk about is developing resistant populations for restoration. And it really doesn't matter what tree species you're talking about. I will advocate to a pretty strong extent, you have to treat trees and other wildlife species different than crop species, okay? Because our intent is quite different. Our level of resistance probably won't be as high. We value genetic diversity out there in the wildlands. We value the adaptability between different areas out there very highly as well, too. So our approach is a little bit different than crops. Same principles, but the approach is a little bit different. You see on the photo here, our white pine blister rust, Carnartium rubicola is the fungus that causes it. It has an alternate host, okay? If there were only pine trees, it affects pine trees seriously and can kill 99% of some of our 5-needle pine trees, okay, in some areas and whatever too. And back in the 40s and 50s, there was research done. They did all sorts of things, chemicals. They tried to eradicate the alternate host, which is a native species, which nowadays is not real favorable to do either. You know, getting rid of one species to save another species and stuff like that too. And so really they came to the conclusion that genetic resistance was maybe the primary path to pursue. And a lot of that's been ongoing at our facility since 1966. You know, I haven't been there the whole time, but it's been ongoing. So kudos to our U.S. Forest Service for funding it over that period of time, because continuity in a program like this, you'll see, is important. But anyway, you can see these trees have cankers on them. They're sporulating. They send spores back to the ribes. The ribes send spores back to the pines. We've got a fantastic system for this fungus to spread. There are 9 species of 5-needle pine in the United States. 8 of them occur in the western states, most of them at mid to high elevation environments. Most of them are very, very susceptible, but that does vary by species. Okay, as you all know, there's new incursions of either pathogens or insects coming in all the time. Certainly this happens in the United States. Genetic resistance is seen one of the primary avenues for getting these species and keeping them back out there. In the United States, the U.S. Department of Agriculture, U.S. Forest Service is the principal agency for kind of like long-term work of this nature. The U.S. Forest Service is broken down into kind of 4 divisions. There's kind of an International Programs Division. There's a Research Division. There's a State and Private Forestry Division, which includes Forest Health. And there's a National Forests Division. And I'm actually in a unit that is not in research, it's not in forest health, it's actually in the National Forest System. So it's a really odd unit. But what it does, it makes us emphasize the practical need to get materials back out in the forest, okay? Publications don't really count to me. I can publish if I want, but it's really, hey, am I getting the job done? Are we getting the materials back out? So what are the threats? You know, we all know that these exotic insects and pathogens have increased public awareness, that the species can go either extinct or become very uncommon. and there's all sorts of associated changes in the ecosystem that happens because of that. That can be complicated because of climate change. But I like the statement that I saw in this one paper about the non-market value of trees from carbon storage, et cetera, far exceeds the commercial value in wood products, et cetera, too. And I think we have to have a greater awareness across society, across our managers, across those who fund programs, that, hey, just because we aren't producing a commodity per se doesn't mean that the loss of these species does not have tremendous value. And so I think, at least in the United States, there's more and more recognition of that, and I certainly state that more and more. I do work with some species that have somewhat commercial value, but I work with a number of species that really have no commercial value. Again, here's a paper, Drivers of Forest Pathogen Invasions, the roles of global trade and climate change. And really, it's the last line I want you to see. There's more and more recognition that even with this happening, that, you know, there's a lot, plants, including trees, have a lot of genetic variation. You know, I'm not sure I know of any plant really that doesn't have some resistance to whatever is attacking it, at least for forest trees. It may be relatively little resistance, you may have to breed it, et cetera, but blanket statements that there is no resistance, even in American chestnut, are just wrong. So resistance breeding programs, they may be, in some cases, almost the only feasible solution for really keeping a species present. You know, first of all, you want to exclude new pests and pathogens. You want to eradicate if you can. You know, if there's biocontrol or microbiota can add to that, that's great. Silvicultural management in some cases may help. Monitoring helps you to see, you know, where these things are going. But if you can harness a genetic resistance, you know, it's natural, it's organic, it's what plants and trees have done those over eons, okay? And even humans have been involved in these activities for, you know, thousands of years, one way or the others. We're the ones that have upset the balance here by allowing new invasions to come in. So, you know, maybe we have a little bit of role to help the plants or the trees to rebalance the equation so that they can continue to exist. So myrtle rust, what will it take? 5 or 6 years ago, I was asked to go over to Hawaii to speak on ohia and rapid ohia death. And I showed a slide a little bit like this. I've enhanced it a little bit, but one of the first things I said, you better find some people who are really passionate and are going to drive any program that you have, because that's what it takes. You know, it's not a research project that's a 2 or 3 year project and stuff like that too. If you really want to sustain these species in the presence of these exotic pathogens or insects and stuff like that too, you're going to have somebody that really focus, and you're going to have public support, and you have to build that in many cases, a sense of urgency. You know, if you can do something in 10 or 15 years, you don't want to spread it out over 50 years and stuff like that too. You know, certainly reviewing other successful programs. Nobody wants to reinvent the wheel. We don't have time, we don't have money, we don't have the personnel and resources to do that. So, you know, really you want to be on the fast track to begin with. You want the proper structuring and staffing of programs. You know, I've been involved for almost infinity in resistance, so I've seen kind of what it takes. I've seen programs that haven't been successful. You know, we have some of the more successful ones out there. You know, continuity of funding personnel. A series of 5-year plans or at least reviews is not a bad thing. I don't mind having a review. I want to make sure my programs are moving forward. And with reviews of that type, often comes support if you show that you're actually moving forward. You know, I mentioned that there used to be a Rust Busters group that in the early stages really helped. And then, you know, I gave a talk in Scotland back in 2015, and one of the slides I showed was a slide that, this photo here, and it's mostly of some technicians, both from our facility and a facility down in Northern California. And I said, you know, it's really a bunch of the key technicians that do a lot of the key work and stuff like that too. And so having that continuity of some key staffing and technicians is really, really vital. It was neat because there was some press in the room. They came up to me after the thing and said, what are the names of those technicians? And they put it in their article and stuff like that. So I thought that was really great because a lot of times, you know, some people like me are the front people of a program. But, you know, I really rely on our technicians and some of the other staff to make this happen. A diagram like this is key. if you're going to move forward, and so I show this at a lot of presentations, and you could develop your own or use something like this. There are really 4 components for making a resistance program, or maybe anything, successful. There's research, okay? So research is valuable. I'm going to put a caveat out there in a minute. And there's kind of tree improvement or tree breeding, and kind of that's where I come in, the applied nature. But there's also management commitment over time. These programs aren't going to happen if you don't get management commitment and funding and whatever too. And then there's the end point, the land managers that put things back out in the forest. As I say, if research does something and nobody's doing anything, hey, I as a taxpayer, do I care for the most part? If I'm breeding the best resistant stuff in the world, but nobody's using it, hey, is there a point to it? I've now, with one of our species, Porterford cedar and the root disease, kind of the 3 of these elements have done their job. And I spoke to the land managers. I said, now it's up to you all. If you start planting the material, and they have, I said, this could be the first species affected by a non-native pathogen that is effectively restored. But it's not in your court. And one of the caveats I have here is that all these components are important. But I would not let an operational applied program get caught up in research. It's not about publications, it's about getting materials out. So that would be my one caution. There's 2 major components or phases to developing resistance. The first one, you ask a bunch of questions. Is there genetic resistance? And, if so, what's the frequency? What's the geographic distribution of resistance? We heard a little bit of that yesterday, so that was good to hear. What type of resistance? Is it complete or partial resistance? Is it due to a single major gene, or is it more quantitative in nature? And that can vary by species. You can even have both types within a species. What level of resistance? Are you ready to go? Our most susceptible parent trees or families have 0 resistance. They all die in our test. Okay, so what level above that do you have, and what level do the managers need before they're going to plant it out? Like I say, this isn't a cultivar where we're going to have 100% of our seedlings resistant or survive. That's not going to happen in most of our tree species unless we use major gene resistance. And I would, in many cases, especially with rust, not advocate using only major gene resistance. Is it durable? Your pathogen or insect has a capacity to evolve. And a long time ago, I think it was Johnson that coined the word durable resistance, and it was you take a cultivar, you plant it out over many hectares, many years, in an environment conducive to the pathogen, and it's durable until it's no longer durable. And if you think about that, they really didn't have an understanding to what makes for durable resistance. Well, it's one thing in crops where you can plant each year. For forest trees where you're planting out there, and they have to be out there for decades or hundreds of years, and they have to be the progenitors of future generations. When we talk durability, we mean durability. That resistance better be durable. We don't want to have to replant or restore every 10 years with new material, and we probably wouldn't have new material anyway. Stability. Is the resistance stable? That's different than durability. Is it effective over different environments? If you read some of the literature, sometimes, and especially with some rust pathogens, a couple degrees shift in temperature, usually upwards, can make resistance less effective. So, I now have some trials going on at my facility where we're testing our resistance under a couple different temperatures at just a few degrees difference, and there does seem to be an effect there with there being less resistance. So with global changing, I want to make sure when we're sending stuff out to the field, is it going to hold up? So these are all questions to ask as you get it out there. Are there fitness costs? And now there might be. I don't think they're huge or anything insurmountable in our case too, but it's certainly a question to ask. You could have a case where, hey, you're developing resistance, there's a fitness cost to the host, and you have a tree that's not going to really grow like a tree or something. And then how to effectively set up a resistance program. So that's phase 1. Is there resistance? How much is there? What level? Et cetera. Phase 2 is, okay, hey, we determined there's resistance, now we're ready to go forward. So do you need to do breeding, or is there enough resistance at a high enough level that you can use some stuff immediately? And it will vary by species in our program. For whitebark pine, some of our populations have enough resistance that we can say, go back to parent tree, collect from X number of parent trees, start your restoration program. For western white pine and sugar pine, 2 of our other 5 needle pine species, the resistance level is so low that you can't do that. You better put that into a breeding program, put them into seed orchards, wait a decade or 2, produce some seed, and get it back out there. How do you organize a program? Large scale, if you're going to go into this, focus on developing resistant populations, not research. You do need infrastructure and personnel. Genetic diversity, you want to have that. If somebody asks, well, how many clones or parent trees you can put in an orchard, I never give an answer. Other than, as many as you can. And it would depend by species how many you're going to find and whatever like that too, because you do want to know what the current level of genetic diversity is out there and can we do something that mimics that. How to maximize restoration, even if you have resistance, the question with whitebark pine now, this is a wide-ranging species. And so there's a whitebark pine restoration national plan being developed. And I think the strategy, hey, can we put on all land bases maybe on 10% of the hectares that had whitebark pine, can we restore 10%? And that's a big challenge to get stuff out there, even when you have resistance and everything too. And then how can genomics or biotechnology aid conventional, breeding? Good questions, and sometimes there's a lot of hype out there. And okay, this is our facility, Dorena Genetic Resource Center. It's near Eugene, Oregon, just south of there. And a couple things you can notice. Down on bottom left and bottom right is our inoculation chamber where we put seedlings in there. It's a fog chamber. I tell people it could be the world's largest fog chamber for plants and stuff like that too. We do things on a big scale. This isn't a research operation. This is meant for operational applied resistance development. And, we can inoculate more plants than all the research units in the United States put together, and because we do things on a big scale. And then in the center bottom, you see a series of our trials. We have many more than that too, but those are trials that we grow seedlings for 2 years for these 5 needle pines. We infect them with the rust, and then we monitor them up to 5 years. We could stop when we find the first resistance, which is usually major gene resistance and stuff like that too. But we want to know what are all the traits or components of resistance that exist, because we don't know which ones of those or which combinations of those are actually going to be durable. So rather than stop at the easy find of resistance that looks real good, real sexy, we look for the whole gamut of resistances and stuff like that too. And again, maintaining genetic diversity, maintaining adaptability. We're not going to remove resistant material from Central California up to the state of Washington, even if it was good material, because in many cases, it's probably not going to be adaptable. So again, the 5 needle pines I talk about, we have 9 species of them. One of them, the whitebark pine, is listed as of January 13th this year under the Endangered Species Act in the United States as threatened, mostly because this is blisterous, but also because of climate change, also because of a bark beetle as well too. And I already mentioned it has an alternate host, ribes. Genetic resistance, 2 broad categories. There's a gray area in between them, but there's major gene resistance, which is usually due to a single dominant gene, at least in our case. And 4 of our 9 species in the U.S. have this major gene resistance documented. 2 of them now have a virulent strain of rust documented that will overcome that resistance, and we think a 3rd one, we just found a virulent strain of rust as well too. So that's a warning out there for major gene resistance. We have some parent trees that their families will be 100% resistance. But I like to refer to that as temporary resistance because there's a good chance in your environment this virulent strain will evolve or migrate in, and then you'll have 0% resistance instead of 100% survival. Partial resistance, quantitative resistance, slow rusting. You'll see some examples of that in a minute. This is kind of a diagram, and yesterday people were talking about resistance. In some case, I think they were maybe talking about major gene resistance, in other cases, maybe more quantitative resistance. This is a diagram I like. This is actually out of Porterford cedar and Phytophthora lateralis, but the same thing applies to rust, really. On the left graph, you see 3 lines, and I apologize for the colors if anybody's colorblind, but the top line is our susceptible control, and you can see it goes to 100% mortality very quick. There were like 42 seedlings in each of these families. They all die, and they all die by like day 100 in the trial. The bottom yellow line, you can see, has no mortality even over a 3-year period of time, okay? And at one point, a pathologist asked me early on when we documented that family, he says, your job is done. You just take that one parent tree and you cross it with everything else. And some of you that have maybe some genetics training would have realize that, yeah, you could do that, but maybe you'll carry forth some genes from that one parent tree that aren't diverse enough to tolerate everything else that they will come up with biotic or abiotic events out there. So our goal is not to take that one parent tree and cross it with everything. If we were maybe in a horticultural sense or a crop sense, we might say, well, yeah, we have one that you can plant out there and, you know, for 10, 20, 30, 40, 50 years, maybe it's going to be resistance, and then you introduce another cultivar, but we're not really in the cultivar business. On the right is what is quantitative or partial resistance. Each of those lines is off of a different parent tree. Their families or seedlings off of different parent trees. The 42 seedlings, and you can see the red line up is the one that's susceptible. It goes 100% mortality, but the other 6 or 7 lines on there all take different trajectories upwards over a 3-year period of time, where some seedlings in each of those families, some of them die very quickly, some of them die at year 1, some die at year 2, some die at year 3. Most of those families never even get to 100% mortality. One of them does, the pink one, does get to 100% mortality eventually at 2 or 3 years, but I would argue that that's more resistant than our susceptible control. So in a sense, all of those are resistant families. Some of them are, and then within those families, if I were to do some within family selection, I would take the best families and maybe the individuals in those families that never died in the trial, and you could put those into the seed orchard or breed those and whatever like that too. So, you know, it's comforting, and maybe that's what you'll see. You'll see this case where, you know, some parent trees out in the field die later, or some within family variation. For the 9 species of 5-needle pine that we have here, the column on the left shows a number of families we've tested for Pinus lambertiana, Pinus monticula. We've tested over 4,000 families for each of those over decades, okay? And the level of resistance in sugar pine or Pinus lambertiana is, we're lucky if we get 5% survival in most of those families, ignoring the major gene resistance. And Pinus monticula, maybe we're lucky if we get 10% survival. There's a couple families with survival above that. The white bark pine, that one was listed as threatened, has a lot more promise. It was originally designated as the most susceptible of all the 5-needle pines, and you got to watch your terminology. The parent trees or the families from those parent trees that are susceptible are extremely susceptible. You can almost say the word blisterous and they'll die. I mean, you can put very few spores on them. They all get cankered in those families. But the families that are resistant there's a fair amount of them, as you'll see in a graphic in a minute. You know, some of those have 40, 50, 60, 70, 80, even 90% survival in our trial. We never saw that with these 2 commercial species, Pinus lambertiana and Pinus monticula. And we think it's a quantitative resistance and stuff like that too. So I think we got lucky, and it sounds like from some of your early data you guys are showing, maybe with at least some of your species you'll get lucky. They won't be like sugar pine and white pine where, hey, you get 5 or 10% survival at best. This is just showing that white pine and sugar pine, these are a bunch of the family trials with up to 120 families in them, and you can see the survival of the 2. You know, in many cases, it was never better than 5 or 10% of the average of those open pollinated families. So not very impressive. So what you have to do in that case, you have to say, okay, we're going to have to do some breeding. We might have to breed for 1 or 2 generations, put them to a seed orchard, and then maybe we'll get 20, 30, 40% survival that our land managers can put out there, okay? So it's again, we're not putting cultivar. Here's an example showing if you, so number 79, which is in yellow on the far right, and you look at survival, and 2 trials with 2 different geographic sources of rust, and one trial it had 0% survival, the other trial it had 3.3% survival. Well, there's a number of other families in there, and you can see that they have anywhere from maybe 7% survival upwards to over 70% survival. So some very impressive, and that's the far right-hand column. What the other columns are the components of resistance, and some of the components of resistance are what percentage of the seedlings got cankers, how many cankers per tree did you get, and that varied very widely. The susceptible got a lot of cankers per tree. The other ones averaged very few. how early do you get your cankers? The susceptible families generally get their cankers very early. Some of the resistant families take a year or two later to get cankers. Do you form a bark reaction, et cetera? So there's components to resistance. And what we find with the quantitative resistance is kind of those families with quantitative resistance seem to have these different traits or this array of components. It's not just one thing that they show. Here's a graph that, this is a field trial of western white pine, Pinus monticola, and you see some dead trees and you see some green trees. And you might be tempted to jump to the conclusion that the dead trees, the red ones, are a susceptible control. But in fact, they're the major gene resistant family. Okay, so they should have had 100% survival. And in some areas for a while, they will have 100% survival. But on this site, there is a virulent strain of rust that had evolved. And now this family is these three families, there's three families with major gene resistance. They're more susceptible, actually, than our susceptible control by a little bit. And this is a graph that kind of shows that on the left, you're seeing the percent cankering in the families with quantitative resistance with the susceptible family, and the three green lines at the top are the major gene resistant family. And the major gene resistant families have